Welcome to the Metabolic Link, a medical and science-focused podcast that explores the common thread of metabolism in health and disease. This is where science meets society. Hi, everyone. Welcome back to another episode of the Metabolic Link, here with my co-hosts, Dr. Angela Poff, Dr. Dominic D'Agostino, and Victoria Field. We're really thrilled to be with you today to talk about some interesting research papers, one of which is on ketone bodies. Actually, it's called Ketone Bodies Attenuate Wasting in Models of Atrophy, and the other is titled Effects of the Ketogenic Diet in Mice with Hind Limb Ischemia. Two really interesting papers that have some ties together that we'll be discussing today. But why don't we kick it off here with the first, which is centered on ischemia. And Dom, would you like to lead the way through this paper and let us know?
Yeah, this paper was brought to my attention just a little while ago, and the results of the paper are kind of interesting because the hind limb ischemia is kind of like a good model of ischemia reperfusion, but also for wound healing too, because it necessitates angiogenesis and repair of tissue, and also may facilitate the anti-catabolic effect of ketones too, could come into play there. This particular group was based out of Shanghai Institute in China, and they highlight in the background of the research, sort of the rationale for the research is that the ketogenic diet has been shown to have anti-tumor effects, anti-diabetic effects, also anti-epileptic effects, may improve cardiac function and in relation to neurological insult, but they explain that there's not good data to indicate the effects of the ketogenic diet on blood perfusion and tissue recovery after ischemia. So there's no explicitly stated hypothesis for this particular experiment or particular study, but one would think going into it that they had the hypothesis that the ketogenic diet would favorably improve hind limb ischemia and the repair of the tissue. So I did notice that in the introduction they sort of define the diet by ketogenic diet as one that restricts sugars, and they talk about ketone bodies being short chain fatty acids. So we know that that's not totally correct. So it's possible that they're not super familiar with the ketogenic diet, and of course ketone bodies are beta oxidation byproducts of fatty acid oxidation. So, but not necessarily fats or triglycerides in and of themselves. So they use a wide variety of methods that we're pretty familiar with in regards to looking at different metabolic parameters. They used a glucose ketone monitoring system that we use. The hind limb procedure, ischemia procedure, was very interesting. They do a unilateral femoral artery ligation in the mice, and this is done under anesthesia, and then they do that for after 24 hours and do this ischemia that damages the tissue. And kind of leading up to that, sort of in the intro too, they highlight some of the effects that the ketogenic diet may be working on in regards to tissue perfusion. They did note that calorie restriction diets, and I'm going to come back to calorie restriction as it pertains to this particular study. So they note that calorie restriction diets reduce angiogenesis, whereas calorie restriction diets reduce angiogenesis, and they state that fasting, or reference a couple articles, that fasting induces or promotes angiogenesis by improving endothelial progenitor cells. So, of course, the fasting would need to be done in the context of no calorie restriction. So that's important to sort of understand.
Do you mean like bouts of fasting followed by like ad libitum or normal calorie intake versus like chronic kind of calorie restriction?
Good point. Yeah, chronic calorie restriction, or even so for mice, 1 week or 2 weeks is sort of like semi-chronic, because that could be like a month or two for humans. But the gist of it is that fasting and that calorie restriction can reduce angiogenesis. They just mentioned one article, but there's a lot of articles, and there's a lot of work that was done by the National Institutes of Health and National Institute of Aging, by several groups, showing undisputedly that calorie restriction impairs wound healing in mice, in rats, and in monkeys. So there's some research that maybe they were not fully aware of, but they're kind of thinking along the lines of, the ketogenic diet would improve various metabolic markers and other things that could potentially enhance, repair and tissue injury in this particular model. Like I said, they do not state the hypothesis up front, but that's what you think of when you're going into kind of reading this study and how it was designed. And the details of the ketogenic diet are not completely clear to me. It's not in the actual publication. There is a supplement that kind of vaguely describes their ketogenic diet formulation. I had mentioned in the past that the diets that we typically use that are ketogenic have MCT, so they further sort of increase ketone levels. But they did use the Doppler perfusion measurement to look at blood flow, and they did a wide variety of staining and immunohistochemistry, looked at RNA procedures from the gastrocnemius muscle to look at different proteins and signaling associated with the intervention. So just to quickly get to the results, so they did, for the protocol, they did a standard diet versus a ketogenic diet, and it was just 2 weeks. So 2 weeks, after 2 weeks, mice underwent a hind limb ischemia, ligation of the femoral artery to do a unilateral femoral artery, and they were given access to 2 different diets after the femoral artery occlusion. So standard diet, standard rodent chow, and their formulation of a ketogenic diet, and these were done in C57 black 6 mice. So they observed that the ketogenic diet induced weight loss and affected metabolites, blood levels in the blood in the tissues. So they describe measuring blood glucose and ketones at random time points, which was kind of interesting. So they and literally it says random blood glucose and blood ketone measurements were made, and using a blood ketone meter over the 4 weeks period during the diet and during the refeeding. So this refeeding scenario has been studied quite extensively as it pertains to stroke or any certain interventions. And so the ketogenic diet mice lost weight significantly in the 2 weeks, and the blood glucose gradually decreased in the first 2 weeks and then remained stable for the 3rd and 4th week. And if we go to blood ketone levels, actually bumped up to 2.7 millimolar during the first 2 weeks, and then maintained 2 millimolar, which is really high for rodent. It's hard to get mice and rats into the state of ketosis. So they're not using MCTs, they're not using any kind of ketogenic agents. So for mice to be 2.7 to 2 millimolar,
it's like severe caloric restriction.
So it's calorie restriction, but I think we can undoubtedly say that they're also, by virtue of implementing a ketogenic diet, that these mice they definitely did not find it hyper palatable, because they lost weight and during the first 2 weeks, and their blood glucose, under the standard diet, their glucose levels were around 10 or slightly above 10 millimolar, whereas it was half that. So their glucose levels were literally half with the ketogenic diet, and that was maintained after the ketogenic diet was discontinued, because they just did the ketogenic diet intervention for 2 weeks. So there was this period of sustained calorie restriction for 2 weeks. So what I think we're observing here, and it was clear in the literature, is that a calorie restriction undoubtedly impairs wound healing through just nutrient deprivation, reduction of IGF-1, and reduction of various growth factors that are needed for growth and repair, including amino acids. Things like leucine and branched chain amino acids actually have been looked at for enhancing repair and injury. So beta-hydroxybutyrate also, they observed that in the tissues, they measured in the tissues, the heart and the muscles were elevated, whereas the liver, there was no elevation of beta-hydroxybutyrate in the liver, which may make sense because the ketones spill into circulation. So that was, but they did notice a couple things that kind of came to my mind is that, for one thing, this ketogenic diet experiment on hind limb injury was really a calorie restriction experiment. That's pretty clear from the body weight changes and also the blood glucose. We see blood glucose trend to go down, but not completely cut in half or more.
Yeah, I will say, we've done a lot of ketogenic diet administration in animals, and blood glucose dipping that low in a prolonged ketogenic diet experiment and ketones getting that high with a ketogenic diet, I can say I've never seen, unless calorie restriction was going on as well. And I do think that's a great point, and that's what makes these kind of studies hard to do. Unless you've had a lot of experience with how to actually to implement ketogenic diet in rodents, it's very, very easy to do it in a way where the animal does not adapt and actually find the diet palatable and actually consume it and maintain calories and not have these kind of potentially negative, body composition changes and things like that. So I agree. I think that there was pretty significant calorie restriction going on in these animals.
They also observed that both beta-hydroxybutyrate dehydrogenase and succinyl-CoA transferase, or SCOT enzyme protein levels were decreased in the limb tissue in mice before and after surgery in the ketogenic diet group. And
so those are ketone utilization enzymes.
Yes. Yep. Ketone utilization enzymes. So, and impaired ketone utilization in the tissue that was more negatively impacted by the intervention, which was the ligation of the femoral artery. So there was more damage in the tissue. But basically saying that that damaged tissue had less of a capacity to use ketones as an energy source, which is pretty interesting. I think if there's one thing that's kind of novel about the study, it's that maybe tissue injury, but I would not predict that. I would think, tissue injury, well, maybe, because ketone metabolism is a mitochondrial-based energy, and if you are creating hypoxia, ischemia, you're damaging the mitochondria, and then you are impairing mitochondrial ATP production from oxidative phosphorylation substrates, which would be fatty acids, although I think fatty acid oxidation did increase in that, but not ketone levels. So you're saying
in that scenario, maybe there's a glycolytic shift happening because of the injury. You have less blood flow, less oxygen reaching the tissue. Naturally, that tissue will shift towards a glycolytic phenotype when oxygen is no longer available for oxidative phosphorylation. Yeah.
So, and there was, looking at the GLUT4, I think GLUT4 levels went down. So this is actually, one would think that glycolysis would be impaired, but GLUT4 levels go down significantly.
That's glucose transporter.
Yeah, glucose.
Skeletal muscle.
Yep, glucose transporter type 4, GLUT1, and hexokinase 2, which is upregulated. It's a major glycolytic sort of rate-limiting enzyme for glycolysis. PDK1, that went up, PDK1. So that's interesting. PDK1 went up, but fatty acid oxidation, on the other hand, actually increased significantly. Fatty acid oxidation enzymes, or CPT1, which is, and then I think CD36 is a regulatory mechanism for CPT1. So they're, very interesting and divergent changes in metabolic biochemistry and enzymes associated with this intervention, ketogenic diet versus standard diet. And I think that's sort of interesting to note. So they saw worse muscle atrophy. Exactly.
Right?
Yes.
With the ketogenic diet.
And getting back to sort of the outcomes, we talked about the metabolic but the outcomes, the fibrotic area was significantly higher with the ketogenic diet. Relative protein expression, so indicative of impaired protein synthesis, and gene expression was influenced. So I don't even know what COL1A2 is, but
I can't remember.
yeah, but the histological and the immunocytochemistry data indicates that there was an impairment of protein synthesis and an increase in cell death that was not recoverable with the intervention.
And
with
they
the ketogenic
saw
diet.
they saw increased inflammation
Yeah,
with the
so
ketogenic diet, which is
Rats were
very
stressed out, or mice were stressed out.
very, we don't. There's a lot of
things.
research showing very consistently reduced inflammation.
Right. So
Now,
They mentioned that, but okay. But one thing,
look,
the IL-1
I noticed
beta
that too.
and IL-6 and IL-18 are all significantly the gene expression for these inflammatory cytokines are significantly reduced. So there's a disconnect between what they're saying in the paper and the data that I'm looking at, and I'm trying to. And that's at the 28-day time point, I believe. So because they're not really clear on what specifically that time point is, but there's a statistically significant decrease in gene expression for IL-1 beta, IL-6, and IL-18, but no doubt that with the ketogenic diet, there's a massive increase in necrotic tissue area. Without a doubt, no one would argue that, and that's super robust if you look at it. Even look at they have some representative pictures of the animals and the limb, and no doubt that the ketogenic diet animals did not do well. But I would argue, and I think it's very clear in the data, although they don't cover this in the discussion like they should, the ketogenic diet was what they're studying here is calorie restriction and nutrient deprivation, which has been validated in numerous models, including non-human primates, to impair wound healing. So I think that's what we're kind of looking at here.
And how much I know you're talking about caloric restriction possibly causing some of the results that we're seeing here, but how much of the weight loss in and of itself might be resulting in some of these, findings? Just when you this is maybe a very broad example, but you see a significant weight loss in somebody who's obese, you might see an artificial rise in cholesterol, things along those lines of how the weight loss in and of itself might have been playing a role here. I'm curious your thoughts on.
With the blood glucose measurements, absolutely, that's the first thing I look at. So the blood glucose measurements, as Angela noted, the only way to achieve that really is through a ketogenic diet that's significantly calorie restricted. And for reasons I don't know, probably because they know that if they put body weight data in there, I think it was very clear that this was a calorie restriction experiment. But the blood glucose data sort of shows that. And their ketogenic diet, I believe, was also not only calorie restricted, but also protein restricted. So you have a protein restricted ketogenic diet that's calorie restricted. So
it's a recipe for worse
odds
wound
are against
healing.
you.
Yeah. So in the context of wound healing, as you know, this would also apply to burns, where it becomes a hypermetabolic state. And a nutrient deprivation in a hypermetabolic state will significantly impair wound healing. And I think that's an important lesson because, a student, Dr. Shannon Kessel, did her research under me actually studying ketone supplements. And I think this data kind of reinforces the need for a ketogenic intervention, I think, because it's going to be hard to put someone on a ketogenic diet that is kind of recovering from injury. So implementation, we know, is very hard. But in the context of a standard diet with a ketone supplement, we saw significant increases in blood flow through Doppler measurements and an enhancement of wound healing in a pretty elegant wound healing model, ischemia versus non-ischemia. And there was trends, or remind me I think it was significant, even we looked at aged animals and also in in young or juvenile and enhancement of wound healing.
Right,
So
and
that's
animals
with ketone
that are not
supplements.
supposed to have impaired wound healing, right? She saw at least some trends, and I think there was some significant, data there that ketone supplementation on top of a standard rodent chow could support. And I do think that what you're talking about makes a lot of sense in terms of like keeping in mind that there are going to be certain scenarios where one ketogenic strategy might make sense more so than another. And, you know, individuals can lose significant body weight on a ketogenic diet. Yeah. Some people do. And there might be, you know, situations where something like a ketone supplemented, you know, formulated whole foods, but more standard type, you know, diet could make sense. And it's sometimes hard. It's hard in this kind of research. Diet research is really tough to design properly. Yeah. And it's tough to carry out. And there's just a lot of these small little details that fall through the cracks in terms of like if you read this without kind of knowing a lot about you know, how like calorie restriction in animals and what a ketogenic diet in mice looks like that's not calorie restricted versus what is, it could be hard to interpret. And
I just want to add that my original hypothesis was when we were discussing her dissertation project, and on her dissertation committee was Dr. Lisa Gould, who is president of the Wound Healing Society and very knowledgeable. Sort of my lack of understanding, which showed in our initial discussions, was that calorie restriction would enhance wound healing. And she directed me to the literature that was not the case, especially when you have calorie restriction and protein restriction. So one of the missing things here not discussed is that you want to get ample amounts of protein. Any kind of protein restriction is going to impair wound healing, and protein restriction will lead to nutrient. So protein is a really key factor, and people have published research on branched chain amino acids, including leucine. Leucine in and of itself, you know, enhancing the wound healing process. There's some data to indicate that. But calorie restriction could the only scenario where I think calorie restriction could, if someone is grossly overweight and their metabolic parameters are really way off, you know, some form of restriction and bringing the blood glucose down to enhance perfusion could help. But that was part of the reason that we didn't do the ketogenic diet or calorie restriction. I was quickly talked out of that. And then, we were doing ketone supplement research at the time, but that was a very powerful intervention. And we need to get that research published. We got a dissertation published, but I think this is a super important topic. And I think it's underappreciated, the power of metabolic interventions for improving wound healing, in this case hind limb, you know.
And I think it also points out that, like, if, you know, trying to think about translating to humans eventually, this sort of dietary therapy needs to be done very intentionally with a very well formulated ketogenic diet, making sure in certain scenarios it's going to be really important that an individual maintains calories, for example, or in some cases maybe cancer patients, that there's a lot of research on ketogenic diet and cancer, and certain cancer types. Cancer cachexia is a very serious problem, contributes significantly to morbidity and mortality, and maintaining healthy body mass is very important in those patients as well. And implementing a dietary therapy, even though it's a diet and everyone has to eat a diet, you know, implementing it with that in mind and understanding that it is a medical intervention, not just changing up your diet.
In the context of this, also enhancing and augmenting skeletal muscle protein synthesis is going to be super important, and that would necessitate high protein and maybe maybe normal levels of carbohydrates, you know, and insulin to augment that wound healing process for someone that's in a hypermetabolic state. So if someone's obese, type 2 diabetic, that could be a little bit, but I think it sort of opens the window for a more formulated and personalized metabolic approach for wound healing. So I'm thinking a lot towards diabetic wounds. And I think that led to the discussion initially with the idea for diabetic wounds, you have someone who's obese, high blood glucose, then calorie restriction should work. But even in that context, the data is not really solid on that. So I think it's really designing a diet, the macronutrients, to really optimize it, but without the restriction. You don't want to nutrient restrict.
Yeah, and just being intentional about, in that situation, address the chronic wound first and then address the underlying metabolic dysfunction that is making them susceptible to that kind.
Can only imagine
condition.
that process of getting a patient who's like got a chronic, like a terrible injury or something along those lines that then has to implement a lot. I mean, it's hard for any sort of patient. But I think that's also that concept is maybe a barrier to entry of starting a ketogenic diet with a cancer patient for fear of cachexia being an issue already. And I think causing 20% of all cancer deaths to begin with, like knowing that the ketogenic diet has the potential to, cause some weight loss, especially if not formulated properly, is a barrier that we need to get past. Because I do think there are potentially ways that you can work around that just in working with cancer patients myself. But it's very complex and it's a dance, and you have to be very, very careful because many of these patients are fragile to begin with. That it's a tough process. And that leads us into the next paper that we have here by somebody who we know quite well, Dr. Andrew Gutnik, titled Ketone Bodies Attenuate Wasting in Models of Atrophy. And this is really interesting following a paper like that and just kind of diving into how exogenous ketones might be helpful when looking at something like ketone or cancer cachexia and muscle atrophy. If you want to kick it off for us,
so
Angela.
This was actually a paper that we kind of decided we should also talk about this kind of at the last minute since we were doing this other hind limb ischemia paper, because it's very relevant and it highlights how complicated science can be in terms of both the results that come out and then interpreting individual articles within the context of the literature as a whole. And I think that's something that can be really hard to do, especially if you're not, in that niche area of research and very knowledgeable about, what's happening in the literature as a whole. And so Dr. Gutnik did his PhD in our lab, and he did a lot of work characterizing a novel model of cancer anorexia cachexia syndrome. So cancer patients, late-stage cancer patients, will often develop cachexia, which is basically the wasting of body mass that is driven by both the cancer itself and also driven by the treatments can elicit cachexia, but it is also driven just by the cancer itself. And so this is a normal kind of thing that happens, but it does contribute to cancer death significantly. And it's hard to treat. Once someone has entered this kind of state, it usually has a pretty grim prognosis.
Yeah. I'd also like to add that the wasting with cancer patients are sometimes multifactorial. And what Angela is going to discuss here and highlight is an intervention that we think may have functional effects on different reasons for the muscle wasting. There's age-related sarcopenia. Most cancer patients are older. There's the cancer cachexia by itself is due to hypermetabolic, hyper-pro-inflammatory state, right? So there's age-related sarcopenia, cancer cachexia, and then chemotherapy-induced cachexia or muscle wasting. So
and anorexia, which comes along with this state.
And that's
which exacerbates
that was highlighted in the paper too. So so anorexia, cachexia, which is hypermetabolic, pro-inflammatory state. Cancer happens in an older phenotype, so age-related sarcopenia, and then you just have an exacerbation, and then in the context, we didn't study standard of care here, but chemotherapy is a massive hit to your protein metabolism and skeletal muscle. So usually you have a negative synergy. You have kind of a worst case scenario of muscle wasting with a cancer patient. So it's very promising that a non-toxic metabolic intervention like this could hold promise.
Yeah. So what was interesting in Dr. Gutnik's paper, which came out of the University of South Florida, so he was basically looking at a ketone ester, which is a source of exogenous ketones. So instead of consuming a ketogenic diet and asking your liver to make its own ketones, ketone esters or other ketone supplements are where you consume either ketones themselves and they go into circulation, or a precursor that gets converted into ketones. And so it's a way to induce ketosis without the strict dietary changes that come with a ketogenic diet. And he was looking at this ketone ester, specifically the RS-1,3-butanediol acetoacetate diester, which we've studied extensively in our lab, in a couple of different models of atrophy. So one being this cancer anorexia cachexia syndrome, and he used a novel model of that. He actually characterized this as the VMM3 mouse model as a really great model system for studying cancer anorexia cachexia, because it recapitulates almost all of those features that we see in cancer patients in the clinic. So he applied ketogenic diet to these animals. And he saw that there was a non-significant but small reduction in tumor burden and ascites, which is fluid accumulation in the abdomen that occurs in late-stage cancer.
And maybe explain, what that is and what that's because ascites, the fluid formation, which happens in end-stage cancer, is associated with tumor burden. Mm-hmm. But I think it's a little bit like... remember, it's kind of, there's a little bit fuzzy understanding as to why the ascites occurs. What I've seen in blood work is that albumin levels really tank, and albumin levels are sort of play a huge role in fluid movement and edema and things like that. Mm-hmm. So I think
and it's also associated with liver dysfunction and inflammation.
Yep.
So. Yep. And so these animals, as they get very sick, they have extensive tumor burden. This model in particular is a metastatic model and has metastatic spread throughout the body, including the liver. And so the ketone ester in and of itself had a small positive effect or beneficial effect in terms of actually addressing the disorder itself. But what was really interesting was...
And no ketogenic diet.
No ketogenic diet. So it's just
clarify, this is therapeutic ketosis with a standard diet. Mm-hmm. We're not suppressing glycolysis, blood glucose, or anything like that. So, and it's having a mild anti-tumor effect.
Yeah. But they did see a reduction in the anorexia. So these mice were not exhibiting this failure to consume calories as normal. They also noticed a reduction in the muscle atrophy of several tissues. So they looked at a lot of different tissues in the hind limb, which was the exact, hind limb in this other paper was studied as well. And in this paper, they saw that this ketone ester prevented atrophy of the gastrocnemius, the soleus, the quadricep, and the tibialis anterior, these muscles in the hind limb. And interestingly, it didn't inhibit fat loss. So this is important because as Victoria mentioned, there's a fear about, and a very valid concern, that losing weight in an individual who's maybe already compromised with cachexia, that could be dangerous overall. And in this study, at least, and there's been other studies and some clinical kind of findings that support that a ketogenic diet in this setting can help preserve muscle mass. And you still might see fat loss, like body fat loss, but muscle, which is really the key thing that we're concerned about. When a cancer patient has cachexia, the biggest concern is that skeletal muscle, or not necessarily just skeletal muscle loss. And fat loss is not as concerning. It's really that muscle mass loss that can be very dangerous.
Functional consequences, lack of independence is once that happens and they can't move, then things go downhill very fast.
Yeah, so this is one big reason why I think ketogenic therapies are, kind of slow to get into kind of the arsenal and in clinical trials for cancer. And I think it's a very valid concern because, exacerbating cachexia could offset any therapeutic potential benefits. But it's interesting to see, as these studies are being performed, that maybe there's actually maybe it's not detrimental in the way that is seriously concerning in terms of preventing or exacerbating lean mass. Thankfully, at least so far, a lot of the data doesn't seem to suggest that. So he showed this in this VMM3 model. So this is a mouse model of cancer-induced cachexia. So ketone ester was able to prevent atrophy in this setting. And then, he asked the question, well, could this be applied to other conditions that are characterized by atrophy as well? And so he also studied this in an LPS-induced atrophy model, so a sepsis inflammatory-based atrophy model. So application of LPS causes muscle wasting, body weight loss, an atrophied state in animals in a very rapid time frame, within
like a
days.
cytokine storm,
yes.
essentially. A very pro-inflammatory state, and those cytokines are highly catabolic. TNF-alpha, the original name for that was Cachexin. So because TNF-alpha Cachexin was renamed TNF-alpha. So it is a primary driver for cancer cachexia. So that was one of the things, looking at. So mitigating the pro-inflammatory pathways are sort of a key pharmacological, interventions are targeting this. And that's kind of sort of what was going on here.
Yeah, very similar. And so he did see that in this model of atrophy, so something very different. I mean, it's a completely separate model of atrophy, that they were able to prevent that body weight loss that is occurring in the model system, and also prevent the anorexia that is typically associated with atrophy. And then they were the ketone ester treated mice had a reduced time to recovery, so they recovered faster than the animals that were on the standard diet. So, and this is interesting because, this study came out of, didn't come out of the blue. It came out of the fact that there's actually a lot of preclinical research looking at ketones specifically as anti-catabolic agents. Evolutionarily speaking, ketones and ketosis in general is thought to be a muscle preserving state. So in fasting, starvation, you have a scenario where the body has to start breaking down protein to be able to release glycogenic amino acids to fuel the brain during periods of calorie restriction or fasting. And the state of ketosis is associated with a slowing of that muscle loss, because ketones, which come from the fat on your body, can fuel the brain. And if you're fueling the brain off of ketones, you don't have to break down as much muscle to support the brain's energy metabolism during that state. And this is why, humans can typically, go much longer without food than they would be able to otherwise. If ketones were not available to be able to fuel the brain, then muscle mass loss in prolonged fasting would be fatal much quicker. So even evolutionarily, ketones are associated with this and known to have this muscle preserving role. And in the past, decade or so, there's been a lot of research trying to understand why. So, and now we kind of understand there are signaling pathways that might underlie some of this this mechanism, such that ketones themselves, through signaling, can inhibit catabolism of skeletal muscle. And so that was why Dr. Gutnik had kind of gone down this path to investigate, and he did see that ketones specifically administered on top of a standard diet were able to prevent atrophy.
And for humans, real quick, there's, at least a half dozen experiments where they give ketones intravenously, and one or two in particular where they gave subjects IV beta-hydroxybutyrate, and which does convert to acetoacetate 2 in circulation, and it reduced the catabolism of branched chain amino acids. So it was shown to be sparing to amino acids, just giving beta-hydroxybutyrate. And other markers have been looked at, like 3-methylhistidine and other markers of catabolism are significantly impacted with ketones.
And that was a really good point, because there is a fair amount of data showing in a nutrient deprivation state that ketones, IV infusion in some humans and also in animal models, is able to prevent that atrophy, which does make it interesting to interpret the results of the first paper, because that is a nutrient. I do think that was probably a nutrient calorie deprivation kind of state, and they did have pretty high ketone levels in those animals, but they didn't see that effect. So something to think about.
Wonder if they threw in ketone supplements in the mix, what would have happened?
Well, I think a big factor there was the calorie restriction and the protein restriction. So protein restriction, that's driving, muscle regeneration and muscle protein synthesis, obviously. There's a higher need for that, and that's not happening with a ketogenic intervention that's has less protein, and then you have calorie restriction on top of that. Whereas the work Dr. Gutnik did was using a standard diet with a ketone supplement where you have, and that would be pretty easy to implement in the context of cachexia or in wound healing model.
Yeah, I feel like both of these papers are really interesting to discuss, both on the ischemia side of things and kind of talking about really the importance of sort of proper implementation and understanding how to implement a ketogenic diet with rodents in particular. It sounds like it's just as complicated as with humans, if not more maybe. They can't talk, so that's challenging. But then also, of course, the potential for application of Dr. Gutnik's paper with cancer patients, dealing with something like cancer cachexia that there's not many options for in terms of, and too, a lifestyle change could be very challenging at that point. So that maybe you have this option. That might also be more comforting for the family around them as well. So really exciting research. And if you want to learn more about these papers, you can find them in the description below. Thank you both for hopping on here, and all of you for listening, for watching. We're going to be doing more research papers, diving deep into the metabolic link and this common thread that ties human health and disease and everything really together, in our opinion. And the research is showing that too. So make sure you guys subscribe, follow, listen in all the places that you have a podcast, and leave us a review for the Metabolic Link, because that's how more people get to know about what we're doing and the research we're talking about. And until next time, we shall see you soon. And thanks so much.
Thank you. Appreciate you watching, listening.